Hello students, once again welcome back to our biology class. Myself Dr. Raghavendra here. Yes, in the earlier class we were discussing about the enzymes and then enzyme activities. Yes, we have discussed about the rate of reaction, enzyme action, nature of enzyme action. We have studied the different kind of factors influencing the enzyme activities also we discussed, such as temperature. At a body temperature most of the enzymes will remain active, and then if the temperature goes beyond 40 degree, yes, denaturing may happen. So there are certain enzymes will be very active even at 80 or 100 degree temperature, and they are mainly the Taq polymerase. Thermus aquaticus is a bacteria found in the hot springs, and the enzyme present in that bacteria is highly active between the range of 80 to 100. That is an exceptional. Well, so temperature also influences the rate of enzyme activity. Other than that, pH. So there are certain enzymes which are highly active at acidic pH, such as pH 2.0 is favorable for the enzyme such as pepsin in the stomach, and enzyme mainly such as a trypsin will be very active at a pH alkaline pH 8.0. Like that, particular range of temperature as well as pH favors the enzyme activities. Beyond that or less than that will not favor the enzyme activity. And then we also discussed about the substrate concentration. Yes, up to some extent, addition of substrate will be increasing the enzyme activities until all the enzymes are get engaged. Once there is no more free enzyme available to act upon the substrate, even addition of extra substrate cannot alter the rate of reaction. So that is the one thing. Other than that, yes, we also discussed there is a correlation between the enzyme activity and the substrate concentration. So more the affinity, yes, there is, we have discussed Vmax. It reaches the maximum velocity of the enzyme activity. We call it Vmax. So at that time, the graph become what we say. It is the rectangular hyperbola. Even you are adding extra substrate, it doesn't alter the rate of reaction. And then the Vmax by half, yes, Vmax by 2 is half of the velocity is attained, and that cuts mainly a substrate concentration value on the x-axis. That is called the Km value. The Km value is mainly a measure of the affinity between the substrate and enzyme, and Km value differs from enzyme to enzyme. Well, that also we have studied. Now, what is the in fact we need to discuss is the inhibition, enzyme inhibition. Well, so inhibitor also influences the enzyme activity. Let us understand that. So enzyme inhibition. Enzyme inhibition is mainly by the inhibitors. There are three type of inhibitors. We can say that yes, it is mainly competitive enzyme inhibition. Enzyme inhibitor, we can say that. Second, non-competitive inhibitor. So third, product itself may be acting as a inhibitor, product inhibitor. So when we say that competitive inhibition, what it is? So mainly under the competitive inhibitor resembles the structure of substrate. Okay, competitive inhibitor resembles the structure of substrate and compete with substrate to bind with active site or substrate binding site. Substrate binding or active site on enzyme. Yes, what happens here, you might have been studied. So this is the enzyme, and Here it's a cofactor. This is what we talk about mainly the active site or substrate binding site. Active site or substrate binding site here. So a substrate want to come and bind with this, and then but what happens, this is a substrate, suddenly so a inhibitor come and closes this. So inhibitor blocking active site. Why? Because inhibitor has close resemblance with that of the substrate. So inhibitor compete with substrate to bind with the binding site or active site of the enzyme. So thus it keep on blocking all those active sites, and it doesn't allow the mainly substrate to bind with. So there is what we can say hindrance in the conversion of substrate to product. So here what you are observing, inhibitor has a close resemblance with that of the substrate. It come and bind with the active site. It blocks that active site. Now the active site no more available to bind with the substrate. So further there is we can say inhibition of enzyme activity. Thus it causes, we can say that inhibitor, mainly the competitive inhibitor, yes, mainly competitive inhibitor reduces the or decrease the enzyme activity. Thus inhibitor compete with substrate, reduces enzyme activity. So for this one simple example, let me tell you, for this example, malonate resembles the substrate succinate, okay, and inhibits the activity of succinic dehydrogenase. Succinate dehydrogenase. Generally, succinate is a substrate that is further converted into fumarate, okay, by the enzyme SDH. But malonate, what it is doing? It is going and binding with the SDH, so SDH cannot do this reaction, which is a very very important reaction in the Krebs cycle. Okay, so this reaction is happening every now and then in the mitochondria, and this malonate is a competitive inhibitor for the succinate substrate. It resembles the structure of succinate and it blocks the enzyme SDH. So an important reaction in Krebs cycle is affected. Well, this is what we talk all about mainly. Yes, competitive inhibitor, they have the structural resemblance. They will mainly what you can say, the structure very much close to the structure of substrate, and they will come and block mainly the active site or substrate binding site on the enzyme. So this active site is no more available for the substrate, so inhibitor block that. As a result, enzyme cannot convert this substrate remain as a substrate only, it never get converted into a product. So that's why the simple example, malonate is mainly very much resembling the substrate succinate. Okay, so what happens, The malonate coming and binding with the SDH and not allowing this reaction, succinate remains as it is. It keep on accumulating in the cell. It is never getting converted into fumarate. Thus, one of the important step in the Krebs cycle is affected by the malonate. This is one essential and important example of competitive inhibition. So, let us understand some other, what we can say, mainly the inhibition, mainly non-competitive inhibition. Let us go ahead with the non-competitive inhibition also. So here in the competitive inhibition, what we have seen, inhibitor is competing with the substrate and affecting the enzyme activity. In the non-competitive inhibition, the inhibitor is not at all competing with the substrate, but it is damaging the enzyme very badly and altering its structure, so that enzyme further will not be, what we can say, available to react. So thus it is damaging or denaturing or altering the structure of enzyme only. That is what we talk about mainly. Yes, non-competitive inhibition. So non-competitive inhibitor won't bind to the active site of an enzyme. Instead it attaches, okay, any other site and alter or modify the structure of enzyme. Further enzyme no more efficient or able to bind with the substrate. So simple example for this, let me tell you, cyanide. Example, cyanide, when it is consumed, it can cause sudden death also. Why? Because so cyanide inhibits cytochrome oxidase enzyme of mitochondria. So when mitochondria itself damaged, there is no chance of survival further. The cell will die very soon. So this is what we talk about a non-competitive inhibitor. So the third category is a product inhibitor. Let me mention the product formed keep accumulating and damaging the enzyme. That is called as product inhibition. What you are observing? Let me tell you, glucose 6-phosphate. Okay, so glucose 6-phosphate getting converted into mainly fructose 6-phosphate. Okay, so here phosphohexokinase, we can say that the enzyme phosphohexokinase. So what you are observing here, basically the reaction mainly glucose 6-phosphate to fructose 6-phosphate is happening, phosphohexokinase is synthesized, but excess of a fructose might or may affect mainly the enzyme in such a way that mainly glucose, we need to understand this reaction, mainly glucose to, yes, here glucose to glucose 6-phosphate, here that is phosphohexokinase, but here mainly what we can say, hexose. So hexokinase, simple hexokinase only. So more of this is an important reaction that keep on happening in the glycolysis. So mainly the glucose 6-phosphate, if it keep accumulating, it can damage the enzyme hexokinase. So more of the hexokinase, yes, will be inhibited or affected by the unwanted excess glucose 6-phosphate, the product, once it is synthesized. Similarly, one more, there is mainly amino acid threonine. Threonine is getting converted into isoleucine. This isoleucine can affect the enzyme threonine deaminase. The enzyme threonine deaminase. So isoleucine is a product that can damage the enzyme threonine deaminase. Thus, it can keep on happening, the accumulation of threonine itself. So thus, product inhibitors are definitely will be there in every cell once there is too much of accumulation of these products. That's why these are the three important main mechanisms of enzyme inhibition. One is competitive inhibition, other one is the non-competitive inhibition, the third one is product inhibitor. Well, I hope you got a clarity about this. This is what we talk all regarding the enzyme inhibition. Well, so we need to go ahead further. The next concept, the classification of enzymes also we will be studying. Fine. So the next, yes, we are going to discuss is all about mainly the enzyme classification. So important concept, enzyme classification, we are going to study. You might have been seen in number of books, they will mention EC 1.1, 1.2, so 1.2.3. So EC refers to Enzyme Commission. So it is the board that decides mainly the nomenclature of the enzymes. So let us understand the concept of enzyme classification. So basically, so far well known enzymes are 6 types. We will be discussing those 6 categories of the enzymes also. Well, so enzyme classification. Yes, enzyme nomenclature. Enzymes are named as EC 1.2.3.4. What is this EC refers to? EC means Enzyme Commission. Enzyme Commission. 1 refers to the enzyme class. 2 refers to subclass. 3 subclass. 4 serial number. Okay, so thus the enzymes are mainly the nomenclature like this they have done. Now there are 6 important categories of enzymes. So let us understand them one by one. The first group what we are going to discuss is called as oxidoreductases. Oxidoreductases. Sometimes they are also called as dehydrogenases. Dehydrogenases. So what actually these mainly the enzymes will do? They will be shifting the hydrogen group or they will be altering. So we can say that they are accelerating the oxidation as well as reduction reactions. So we need to understand what is oxidation. Oxidation, it is loss of electrons. Okay? Yes. So what is reduction? So it is the gain of hydrogen. Well, oxidation and reduction is happening every now and then in the cell. So to consider one simple example as such, there is yes, it is reduced form, plus S dash, it is oxidized form. So the reaction is catalyzed by dehydrogenase or oxidoreductases. What will happen? S is become now oxidized, plus S dash becomes mainly reduced. This is what we talk about mainly the reaction. One more simple example let me give you. A lactate, the compound is converted into pyruvate. The reaction is interconvertible, during which release of yes 2 hydrogen plus 2 electrons. Okay, so yes, here what happens? Mainly the lactate is now what you can say, yes. So here it is a release, yes, it should like this, and then now we can say that here the reaction can be addition of 2 hydrogen plus 2 electrons, gain also. So lactate we can say when it is gaining, it is a reduced form. Okay, so when the pyruvate, it has mainly the lost, so it is a mainly oxidized. So we can say that mainly here it is, this reaction is mainly oxidation of lactate going to form the pyruvate. So the reduction of the pyruvate is going to form the lactate. So here it is a reduction reaction happens. Okay, this is one simple example to say what is oxidoreductase. So oxidation as well as reduction, what we have seen. So gain of hydrogen is reduction, loss of hydrogen is oxidation. Fine. So this is about mainly the first group oxidoreductases. Second group of enzymes, transferases. So transferases are mainly the enzymes that are known to transfer the groups. Okay, so it may be the transfer of the hydrogen or the carbon or the oxygen or mainly amine group. So variety of chemical groups are transferred. Okay, so from the substrate to the yes, mainly S to S dash, we need to know that. So transferases, the enzymes, the group of enzymes known to transfer, okay, transfer the chemical groups. So here we can say that mainly the S has the group G plus S dash. Now after transfer S plus S dash G. So the G from S, it has shifted to the S dash. So this is what we say the transferases. So I hope the transfer of the amine group or transfer of any kind of nitrogen or hydrogen. So these mainly going to convert one amino acid into, or transfer of hydrogen or carboxyl or amine group. Yes, it convert one amino acid into different type of amino acids. Now the third kind, yes, we need to know the third type of mainly the enzyme, they are hydrolases. Okay, yes, hydrolases. So hydrolysis of a compound or by release of the water. Okay, so the enzymes catalyzes hydrolysis. Okay, so we call them as the hydrolases. So they mainly, what we can say, they they convert the groups. So hydrolysis of ether, ethyl, peptide, glycosidic, and then there is what we can say carbon to halide group, and yes, phosphate to nitrogen groups. So the bonds are broken and then release of the water is done, and that happens mainly in the hydrolases. So these are most important, what you can say, the enzyme group, we call them as the hydrolases. So mainly the bonds are broken and release of the water. So catalyzing the hydrolysis of these ethyl, ether, peptide, glycosidic, C halide and P to N bonds. Okay, so this is called as mainly hydrolases. So hydrolases are the most important type of enzymes. The next group we are going to discuss, mainly it is lyases. So lyases also the fourth important category. Lyases, the enzymes catalyze removal of the groups other than hydrolysis. Let me tell you here, carbon to carbon, it has X, it has the Y. Okay, further we catalyze the reaction X and Y plus carbon, double bond carbon. So these are what we can say lyases. These are the enzymes that catalyze mainly the what you can say the biochemical reactions other than hydrolysis. Then yes, we need to understand the one more type of mainly what we can say biochemical reactions that carried out by certain other group of enzymes. We call them as isomerases, and then yes, ligases also. So let us understand isomerases, the fifth category of enzymes. Isomerases, the enzymes that forms the similar kind of type of chemicals or isomers with having or mirror images, we can say it is mainly what we called as a geometric as well as optical. Okay, geometric as well as optical, and then we can say that one more important term as such as the positional isomers. Yes, we need to know that geometrical, optical and positional isomers we call them. So example, yes, glucose 6-phosphate converted into fructose 6-phosphate. It is mainly aldose group and it is the ketose. Both have the C6H12O6 only. Yes, both are the mirror images of one another. Isomerases, we can say. The enzyme, yes. So it is mainly phosphokinase, yes, isomerase. So phosphohexokinase isomerase enzyme. Number 6, ligases. The enzymes link the groups. Okay, carbon to oxygen, carbon to sulfate groups or sulfur, then carbon to nitrogen, phosphate to oxygen. Okay, so these are mainly ligases which link the different compounds by forming the chemical bond. So this is all about today's class. Yes, so little small concept will be remaining that we will discuss later. Till then, take care. Thank you very much.